All right, if you'll recall, last time we identified the four biochemical pathways that are involved in aerobic cellular respiration: glycolysis, the transition reaction, the Krebs cycle, and the electron transport chain. So now we're going to focus in on glycolysis and what's going on in glycolysis. And the first thing to note is where it's happening is in the cytoplasm. So this is the biochemical pathway known as glycolysis. Glycolysis, if you translate the word, means glucose splitting. And that makes sense because if you look at the overall reaction, we're taking one molecule of glucose and splitting it into two molecules of pyruvic acid, also known as pyruvate, right? So glucose splitting, glycolysis. As I noted on the previous slide, it's taking place in the cytoplasm, so it does not involve the mitochondria. That's the point here. Glycolysis, the other reactions require mitochondria. The transition reaction, the Krebs cycle, and the electron transport chain require mitochondria and require oxygen, whereas glycolysis takes place in the cytoplasm and it does not require oxygen. It's anaerobic. And because it's anaerobic, glycolysis is what fermenters use to ferment. So glycolysis and the process of fermentation are very closely connected or related to each other. So as you can see from this diagram, there are 9 reactions in the whole biochemical pathway, and I've numbered them for you, but I'm not going to ask you to reproduce this in any way. But there are 2 phases to this process that you do need to be aware of, or you do need to understand. And the first one takes place during reactions 1 through 4. In other words, that's what we call the investment phase of glycolysis. And what's being invested is ATP. What's being invested is energy. These are endergonic reactions that need to start glycolysis off, and it requires ATP to supply that energy. So this is the investment phase. Then we have reactions 5 through 9, which are referred to as the payoff phase, because as you can see, we're getting NADH and ATP and 4-ATP for a net production of 2 ATP. Because we use 2 here, we're getting 4 here in the payoff phase. So that gives us a net production of 2 ATP through the whole process of glycolysis, which isn't very much. So if you'll recall, NADH is an electron carrier, and those, if there's oxygen, are going to be transported into the mitochondria to the electron transport chain. They're going to deliver their electrons to the electron transport chain. But if there's no oxygen, that won't happen. And they'll have to be dealt with in fermentation. The ATP, as I pointed out when we went over the previous slide, is being made through what we call substrate level phosphorylation, right? So in the payoff phase, the ATP that's being produced is by substrate level phosphorylation. In other words, if you'll notice, as we go from this molecule that has 2 phosphates on it to this molecule only has 1 phosphate on it, that molecule with 2 phosphates, one of the phosphates was taken off and put onto ADP to make ATP, right? And the same thing's happening here. There's a phosphate on this molecule, but there's not one on this molecule, so that phosphate was taken off of that molecule, which would be referred to as a substrate, and placed onto ADP to make ATP. So that's substrate level phosphorylation. So all the payoff here is NADH in the form of electrons and 4 ATPs. The next thing to note is that each one of these reactions, all 9 steps in the biochemical pathway, are regulated by an enzyme, by a different enzyme. And there's just one example that I want you to be aware of, and that's phosphofructokinase, which operates right here. And the importance of phosphofructokinase is that it's the regulating enzyme of glycolysis. In other words, glycolysis is phosphofructokinase dependent. If phosphofructokinase is available or active, then glycolysis happens, and if phosphofructokinase isn't activated or not around, then glycolysis will not take place. So it's the regulating enzyme of glycolysis. And the name tells you what it does. This is phosphofructose. And phosphofructose is getting a phosphate placed onto it by ATP. It's being phosphorylated, right? That's what we call phosphorylation. So phosphofructokinase, you know it's an enzyme because it ends in ASE, and it's an enzyme that adds a phosphate onto phosphofructose. Just as in photosynthesis, what you need to be aware of in these processes are what goes in and what comes out. In other words, what are the reactants and what are the products? So the reactants of glycolysis are 1 glucose, of course, 2 ATP for the investment, 4 ADP, because we're making 4 ATP, that means these 4 ADP are reactants and need to go into the reaction in order to be energized back to ATP, and 2 NAD+. And we're going to ignore the phosphates here. So those are the reactants. The products of glycolysis are 2 ADP, that's these two here, 2 NADH, 4 ATP, that's these 4, but it's a net of 2 because we use 2 here, and 2 pyruvates, or pyruvic acids. The most important products being NADH, ATP, and pyruvic acid or pyruvate. And that pyruvate contains energy. So even though it's the end of glycolysis, that pyruvate is going to go on if there's oxygen for the rest of aerobic cellular respiration, and the energy that's contained within those pyruvates or pyruvic acids is going to be released and used to make more ATP. Likewise with the electrons that are carried by NADH. So I wanted you to understand just the length of this biochemical pathway, and that metabolism involves these long biochemical pathways. But we can simplify this, of course, and that's what this diagram does. So this is a simplified diagram that represents only the investment and payoff phases of glycolysis. And it also shows the carbon chains, right? For example, glucose is a 6-carbon compound, C6H12O6. So there's the investment phase, and I noted on here that that's steps 1 and 3. That's where ATP is supplying phosphate and phosphorylating substrates along the pathway. And then the payoff phase is where 4 ATPs are made in step 6 and 9. And step 5 is where we get NADH. We get electrons harvested by NADH. So as I mentioned, glycolysis and fermentation are closely linked, and organisms that ferment, all they get is the 2 ATP produced through glycolysis. So that seems like a disadvantage, because 2 ATP is not very much compared to what compared to what we end up with aerobic cellular respiration, a total possible of 38, if you'll recall. But the advantage of fermentation is that it can be carried out without oxygen, and it's very fast. The disadvantage of fermentation is that fermenters only get 2 ATP from each glucose molecule, like I mentioned, instead of 38. Total possible maximum of 38 anyway. And they also have to contend with this buildup of pyruvate, so they have to do something with the pyruvate, otherwise the process is going to stop. And they run out of NAD+. There has to be enough NAD+ in order to keep glycolysis going. So those are all problems with fermentation. And fermenters have to solve those problems, but the most notable problem with fermentation is only 2 ATP. But again, it's really fast. So if there's plenty of glucose available, they can make a lot of ATP in a relatively short period of time. And the last point here is that by the end of glycolysis, we have 2 pyruvates that still contain a lot of chemical energy. We still have carbon-to-carbon bonds here, and when those, what we're doing in the whole process of aerobic cellular respiration is breaking those bonds and harvesting the energy from those bonds to make ATP. So there's still plenty of energy left in this pyruvate. There's still plenty of energy carried in as electrons in NADH. And so if there's oxygen, then the process continues where the pyruvate enters the mitochondria through the transition reaction and enters the Krebs cycle through the transition reaction, and more electrons are harvested, and then those electrons are delivered just like the electrons in NADH here. All the electrons from all the processes are delivered to the electron transport chain, and that's where most of the ATP is made. So if oxygen, then on to the transition reaction and the Krebs cycle and the electron transport chain. Those are the aerobic parts of aerobic cellular respiration. And again, the NADH, the electrons are going to go on to the electron transport chain if there's oxygen. So if there's oxygen, there's plenty more energy to be had. So what are fermenters doing? Well, there are two forms of fermentation. There is alcoholic fermentation. That's where pyruvic acid and NADH are turned into alcohol and carbon dioxide, and then, of course, NAD+, the uncharged version of NADH. And then the other form of fermentation is lactic acid fermentation. And same thing, pyruvic acid is turned into lactic acid, just like pyruvic acid is turned into alcohol in alcoholic fermentation. So that solves the problem of removing pyruvic acid, turning it into something else. Because remember, that's one of the problems that fermenters have, is the buildup of pyruvic acid during fermentation. So in this way, they turn it into something else. But that poses a problem too that we'll come back to later. But regenerating NAD+ is an important part of both fermentation processes too, because without NAD+, the processes can't continue. Or what I should say is glycolysis can't continue. So you'll notice here that both processes involve glycolysis. So this is glycolysis, this part of the diagram, just this arrow here. And these are the two ATP that fermenters get from glycolysis. So alcoholic fermentation can be performed by certain species of yeast and bacteria. So, back when we were studying prokaryotes, I tried to relate to you that prokaryotes are capable of just about every form of metabolism that there is. So yeast and bacteria, or prokaryotes, are able to carry out alcoholic fermentation. And lactic acid fermentation, you may be familiar with that. When you exercise, your muscles produce lactic acid, and the reason they're producing lactic acid is because they have run out of oxygen, and they are producing lactic acid as a way of keeping ATP production going, so you can continue exercising strenuously, so you can continue your activity, whatever it is, like running away from a Tyrannosaurus Rex. But again, prokaryotes can do this too. So prokaryotes can do everything. So the commonality between the two of these is that they rely on the reaction, a reaction that follows glycolysis. So the whole point is to keep glycolysis going, but they have to do something with that pyruvate. So the pyruvate is a reactant in both of these, as you can see, but they're turned into two different things. And that's the two different forms of fermentation that there are, alcoholic and lactic acid. So glycolysis is producing pyruvic acid or pyruvate and producing ATP, but both of these need to regenerate NAD+, right, to keep glycolysis going. NAD+ is a reactant in glycolysis, and without NAD+, glycolysis is going to stop, and they're not going to be able to, fermenters are not going to be able to make ATP. So not only do these reactions turn pyruvate into either alcohol or lactic acid, but they also take electrons from NADH and turn it back into NAD+ to keep glycolysis going. So NAD+ is really important to fermenters. And of course, these reactions are regulated by different enzymes. Enzymes run the show. And a reminder, because this was on the previous slide too, the two main advantages of fermentation is that it doesn't require oxygen, and therefore it doesn't require mitochondria, and it's fast. So no oxygen required, no mitochondria required, because that's what this part of the diagram is showing, is mitochondria. So all of this, glycolysis takes place in the cytoplasm. So this is in the cytoplasm, the cytosol labeled on this diagram. And that's where fermentation takes place too. Doesn't involve mitochondria at all. So it can be performed by things that don't even have mitochondria, like prokaryotes. However, it only makes 2 ATP per glucose. That's a problem. And these products are toxic. In a closed environment, the buildup of ethanol will actually kill the organisms that are fermenting it. And the buildup of lactic acid, it is an acid. Lactate is lactic acid, and the buildup of lactic acid will decrease the pH of the environment and kill the organisms that are producing it. In an open environment, it's not a problem because they'll just wash away. They'll be diluted in the environment and go away. But in a closed environment, like when someone is fermenting alcohol to make an alcoholic beverage, the organisms that are producing it in a closed bottle, for example, are going to be eventually killed. And that's why the alcohol content can only go so high. It goes as high as the organisms can survive, and then once they're dead, that's the highest alcohol content that can be produced. So ethanol is toxic, lactate is toxic because it's lactic acid and is acidic. But if there's oxygen, then we go on to the mitochondria and the transition reaction, the Krebs cycle, and the electron transport chain, and the pyruvate goes on to participate in those processes that completely release the energy from that pyruvate. And the NADH produced from glycolysis goes on also to supply electrons to the electron transport chain and produce a lot of ATP. So if oxygen, we go on to the mitochondria. And what's going on in the mitochondria? So what we're going to do here is identify all the different structures of the mitochondria and which parts of aerobic cellular respiration are taking place in each structure of the mitochondria. So mitochondria, if you'll recall, are the product of endosymbiosis 1, or in other words, mitochondria have common ancestry with prokaryotes, once free-living bacteria or prokaryotes. So that's that concept of endosymbiosis 1. And there's a lot of evidence supporting that. They have their own DNA, they reproduce by binary fission, they're about the same size and shape as a prokaryote. So remember that out in the cytoplasm, here we are outside the mitochondria in the cytoplasm, and that's where glycolysis takes place, right? So glucose is split to pyruvate, and that's an anaerobic process, does not require oxygen. Everything that happens in the mitochondria requires oxygen. So we've got an outer membrane to the mitochondria, and that's what pyruvate has to get across, or pyruvic acid has to get across. So does the NADH, has to cross that outer membrane to enter the mitochondria to participate in the aerobic part of aerobic cellular respiration. And those are coming from glycolysis, right? Then we've got an inner membrane, and you can see that the inner membrane is highly folded. And those folds are known as cristae. So each fold is a cristae. And folding in biology, we see folding in biology all over the place, increases the surface area. So this up arrow SA means increased surface area. The purpose of the folding is to increase surface area to be able to carry out more aerobic cellular respiration. So pyruvate and NADH also have to pass through that membrane to get into what's known as the matrix, which is the fluid-filled space, the aqueous solution-filled space inside the inner membrane. That's the matrix. There's more going on in the inner membrane itself that we're going to come back to, but what's happening in the matrix is the transition reaction and the Krebs cycle. So both the transition reaction, once the pyruvate gets into the matrix, it undergoes the transition reaction, which transitions it into the Krebs cycle. That's why it's referred to as the transition reaction, because it transitions pyruvate into the Krebs cycle. There's also high pH in the matrix. In other words, low hydrogen ion concentration in the matrix. And that's going to be contrasted with outside the matrix, across the inner membrane, in the intermembrane space, right? So this is the intermembrane space in between the outer membrane and the inner membrane that has high hydrogen ion concentration. And as you're going to learn, that's the result of proton pumps in the inner membrane that are pumping protons from the matrix out into the intermembrane space. So we get a high hydrogen ion concentration in the inner membrane space, so we have a concentration gradient across the membrane, across the inner membrane, and that's energy. That's potential energy that's going to be used in chemiosmosis to make ATP using ATP synthase, just like we saw in photosynthesis, the light-dependent reactions in photosynthesis. It's the same mechanism, chemiosmosis through ATP synthase. And just like in photosynthesis, what's powering the pumping of protons or hydrogen ions across the membrane is an electron transport chain. Electrons passing through the electron transport chain, electrons being delivered by NADH and FADH2, are traveling through the electron transport chain, flowing through the electron transport chain. That's electricity, powering the pumping of hydrogen ions across the membrane to build up that high concentration in the inner membrane space. And those hydrogen ions then are going to be allowed to diffuse back into the matrix through ATP synthase. That's the process of chemiosmosis, to produce ATP. And because this whole process requires oxygen, it's referred to as oxidative phosphorylation, as opposed to, in photosynthesis, it's photophosphorylation. Here we have oxidative phosphorylation. In other words, putting a phosphate onto ADP to make ATP. So that's what's going on in the mitochondria and where it's going on, or where it's happening.